Malnutrition deaths every day, roughly 24,000 a day. That's basically the same as the number who die every day from cancer. If we can increase plant productivity by combating insect pest and pathogen losses, we would save land and we'd create a lot of new water. So there's human toll and there's environmental toll to plant diseases.
Today we're joined by pioneering force in plant biology, Howard Hughes Medical Institute investigator and distinguished professor at the University of North Carolina, Dr. Jeffery Dangl. Best known for revolutionizing our understanding of how plants defend themselves against pathogens. His groundbreaking research has not only illuminated the genetic and molecular frameworks of plant immunity, but has also had global implications for agriculture, sustainability, and food security. Plants, just like animals, have to defend themselves against a number of forces that want to invade and attack, from bacteria and viruses, just like we do. And so to discover the immune system of plants was such a remarkable achievement that you awarded the prestigious Wolf Prize in agriculture. It's one of the most monumental achievements in all of agriculture for your long-standing work. And so we're thrilled to have you here on National Champion Radio. Thank you for joining us.
Thank you, Andre. It's a pleasure to be here to introduce people to plant diseases, plant epidemics, the plant immune system, and why it really matters that people think about these things. I should point out that the Wolf Prize for Agriculture, I actually shared that with two colleagues and very close friends with whom we've worked on these topics since the mid-1980s. That's Brian Staskowitz at UC Berkeley. He's a noted plant pathologist and molecular biologist, and Professor Jonathan Jones from the Sainsbury Lab in Norwich in the United Kingdom in England. And as you noted, together we were cited for our work in sort of elucidating a model for how the plant immune system is organized and how it works. But of course that work, which I say we began in the 80s, stood on the shoulders of decades of prior science, and it's the product of over 100 trainees, postdocs, graduate students in my lab, and an equal number in Brian and Jonathan's lab. So it definitely takes an entire community to create knowledge at this level.
Yeah, I mean, that part of it, I mean, of course, graciously, thinking about all the things that go into it, it's such a big endeavor to create knowledge and to generate it. Why in this specific field, and how did you get involved in uncovering and discovering plants? You said you sat on the shoulders of giants, and so you sort of, came into a world where this stuff didn't exist, but a lot of interesting things were happening around that time. Maybe you can bring us back and talk about how you got interested and why plants are important.
So I was a biology undergraduate major and an English major at Stanford, and when I stayed there and entered a PhD program in genetics in the early 1980s, I was fascinated with the mouse immune system, and I had worked in a lab. A very wonderful person named Glenn Herzenberg and his wife Leonore offered me a position as an undergraduate researcher in their lab in 1978. And I didn't know doodly when I started. But like all students, I was interested in seeing whether research was a life I could enjoy. And I didn't want to go back to my small hometown in Northern California for a second summer because there were no jobs and it was kind of boring. So they offered me a job, and I grew into that and ended up staying in their lab for my PhD, and I worked on the structure of mouse and human and rabbit antibody molecules. And I think we all know now how important antibodies are in the immune system. And we were one of the first labs. Vernon Oy, who was a postdoc, led this effort. We were one of the first labs that created chimeric antibodies, meaning
give us a overview of that. I mean, just for our young people in the audience. I mean, chimeric antibodies. That sounds ghoulish, interesting. Yeah, how do you get involved in that? And we've sort of heard some of these experiments start to come out, in modern times and their importance.
That's right. So antibodies are exactly like a fork. And the end of the fork that you use to stick into a piece of food is where the antigens bind. And the base, the handle of the fork, is the way that the antibody molecule talks to other cells in the immune system and tells them to do something. Kill the invader, make more antibodies, a variety of things. So we engineered an identical end of the fork to bind a particular antigen. We cloned the DNA for that, and this was when DNA cloning was very young. We cloned the DNA for that and hooked it up to the genes that encode the handle of the fork. And that way we could study the functions of the handle of the fork independent of different combining sites. Because our combining site, the tine of the fork, the antigen binding site, they were identical in all the molecules that we created. And I'll tell you what that led to. Among other things, if you know someone who's had cancer, and I think most of us do, many cancers these days are treated with antibodies. And those antibodies are nearly always engineered to be perfect, to do the job they have to do. And that was some of the baseline work that we did in the mid-'80s gave rise to those therapeutic antibodies 20 years later. So that got me interested in immune systems. Then one day, Vernon Oy, my mentor in the Herzenberg lab, said, look, there's this new paper, you have to go down to the library. That's when we actually had to walk to the library. It wasn't all online. And he said, go find this paper and make us both a copy. So I went, I found the journal, and the journal kind of fell open, and I'm not making this up. It fell open to a page of a different article. The title was something to the effect of specific recognition of fungal cell walls by plant cells and the defense reaction. And I thought, what the heck? I had never taken a plant biology class, still have never taken a plant biology class. But yeah, it's true, don't tell anybody. But I started reading up on this, and I discovered that there was a rich genetics of plant breeding that started in the early 1900s, 1905, describing genetic systems that allowed plants to recognize pathogens specifically. And now I did know that plants don't have circulating immune cells. They don't have a bloodstream the way that we do. So I thought that's fascinating because if the cells are all stuck in place, then every cell in principle needs to respond to every pathogen in principle. So there's no dedicated surveillance system like we have in the animal immune system. So I did a lot of reading, and it was fascinating. That's how I got into it.
that is fascinating, and we definitely want to dive more into those specificities on how we have this surveillance system, immune system that we're used to seeing, even from the Magic School Bus episodes that I grew up on, right? You got the whole body you go into, and of course you got the immune fighting cells. And then in the plant, it wasn't like, they don't have a bloodstream. They don't have, white blood cells to move around in the same way that we do. And so they have every cell reacting to the, pathogen that's trying to harm the plant at the same time.
That's right.
Is that right?
That's
And so
exactly right.
before we dive too deep, taking a step up, thinking about the importance of plants. Right now there's a major challenge going on in the world, and I don't know if a lot of people are aware of the crisis that's happening in the food supply, the necessity for resilience and diversity in our plants. And maybe we can dive into your work and why plants are just so important overall as a high level, and then dive into some of the more specifics.
Sure. So look, plant health is human health. We rely on plants on Earth for all of our food. Every molecule in your body is ultimately derived from photosynthesis, where plants capture light. They turn that light into carbon, sugar, nitrogen assimilated from the soil, phosphorus, all that gets made into proteins. You eat that plant, and your body absorbs those energy molecules. And you build your own proteins, DNA and RNA, and everything else. So without plants, human life would not exist. And
everything we eat is either, Yep.
Yeah, go ahead.
No, everything we eat, like you said, it's either a plant or something that just ate a plant. That exactly is how this system works, and the importance of plants can't be understated.
Sure. So what I just want to describe is the statement that plant health is human health and it's environmental health. And as Andre noted, everything you eat is a plant or ate plants before you ate it. So we go ahead. So on the left, you'll see that since 1970, the population's gone up quite a lot. And on the right, the land use to grow crops on arable land has gone way down. Cities are growing.
So this chart on the left is saying that there's less land to farm on. We're saying
that's exactly right.
And this population is human population.
Right. So we're going to feed what everyone thinks is about 9.5 billion people by 2050. We're going to have to do it on less available land. And there's only two ways to do that. Plow the rest of nature, which I think nobody wants, or get more food off the land that we farm and get more nutrition from that food. So the problem is that all that carbon that photosynthesis makes is not only food for you and I, but it's food for fungi, bacteria, insects, nematode worms. So there's lots of organisms that want that photosynthate, and they cause diseases. And we'll get into in a minute what those diseases are. But if you click forward, this in the background.
That's a huge, just to emphasize it. We see this population going up, and now there's half by 2050 of the amount of land. We lose 10 to 15% every year. Can you just give us a brief view, like what is causing us to have half as much land by 2050 to actually grow and farm on? Is it the diseases that are coming in?
It's the human population growing. They need houses. So the land is shrinking down. And so this is simply one disease. I'll use this at the start as an example of what we can achieve. So in the background are papaya trees that are resistant to a particular virus-like disease. In the front are their parents that were not genetically engineered. They're all dead.
Mm.
So that's the first example done in Hawaii by a Hawaiian guy named Dennis Gonzalez. When he was working at Cornell, he genetically engineered papayas and cured that disease. Now, that's the only place papayas are grown in the U.S. So that's the entire papaya industry.
Wow.
And he saved it. And he's gotten many awards for that work. I'll also note here that you'll see some slides, the lower left, the logo of the Two Blades Foundation. This is a nonprofit group that I'm involved with. I'm a member of their advisory board, and they sent me some of these slides. They do great work trying to implement the results in labs like Brian Staffowitz and Jonathan Jones and others. So at a high level, this is the challenge. More food, more nutrition on less land without destroying what's left of nature. All right, so here's just a few examples that we pulled up out of literature, and I'll mention a few of these diseases later in detail, but there's a thing called wheat rust that has been around since the Roman Empire. So remember, wheat was domesticated maybe 10,000 years ago. So even by Roman times, 2,000 years ago, they already had wheat. They were growing enough of it, and the fungi were right there. There's another thing called Asian soybean rust that moved from Asia to South America. And the notion was 30 years ago, that won't be a problem in the U.S. because it's too cold in the South. Well, now we have global warming, so that disease is moving north because the temperature is getting warmer in the north. So we now have Asian soybean rust along the Gulf Coast. Potato late? Go ahead on.
No. I mean, it's something that I think is getting lost here in this climate change discussion. You know, when you talk about some of these second and third order effects, like diseases moving north that have never been this far north before, impacting crop yields for our population, and it's been a conversation, and I think that often it's relegated to percentages of carbon or some of these other spaces where there's contention on action, right? Where we become stagnated in our ability to move forward and solve the problem. And obviously understanding the importance of our food will, and perhaps the price of it, maybe if there's a drought that, creates or a disease that creates a price increase, perhaps then people will start to understand how important this work is and how important it is to combat climate change at large.
Absolutely. So a couple other diseases. In Africa, people get a lot of calories from cassava, and there's a huge problem in cassava. There's actually a couple diseases of cassava. And then in the U.S., tomato, even here in North Carolina, Florida, upstate New York, there's a disease called bacterial spot disease we'll come back to later. Now, you mentioned a minute ago, prices going up. Click one more forward. Here's three crops that are really in trouble.
Whoa.
And you and your listeners may have read recently that chocolate prices are going way up and coffee prices are going way up. And at least a good portion of that price increase is due to the diseases shown here. And these three plants, cacao, banana, and coffee, also have the problem that at least banana has the problem that it's clonally propagated. And that's something we can come back to later. But
no, I think that's a great. Sorry, go ahead. Sorry.
Go ahead.
I mean, I think that's somebody out there watching. We have to get the problem fixed. A basic way to look at it is coffee's going up, chocolate is going up, and bananas. The price of all three of those things, very vital. Do you like chocolate? Do you like coffee? Do you like bananas? Like all of those are in danger right now because of the catastrophic diseases that are impacting them right now. And so, listen, I could dedicate my life to trying to protect coffee. That's how much I like coffee. So, you know, and I know there's some people who love chocolate out there. And so just on a very topical, high level, simplified level, chocolate, bananas, and coffee are all in danger in the next, how soon are we talking?
Certainly in the next 20-ish years, if not before.
So that they could be eradicated if a solution isn't found.
Certainly banana could be eradicated.
Bananas could be eradicated in the next 10 to 20 years if a solution isn't found to this problem.
But particular kind of bananas that we eat were chosen back in the day because they all ripened in that bunch of bananas that you buy at about the same time, and that they were easily transported. They didn't bruise. So they've been chosen to be the, let's call it the domesticated banana. But they're clonal, so they're highly susceptible to disease. And in fact, the banana that banana replaced was wiped out by disease. So we've already had one, let's call it catastrophic market failure for banana production. There are hundreds of banana varieties, thousands around the world. But they have to have market qualities, you know, in addition to disease resistance.
Yeah. So to introduce them into, you know, growing into the United States, for example, or even transporting them here, they, like you said, those start to make them not efficient in a market sense. Right. Whereas the bananas that we depend on are cheap, they're affordable, and they're widespread, yet they have a single point of failure. Our bananas have a single point of failure because they're all clones. They're all the same banana. The same bananas running around getting cloned all across the United States, and we're all eating the same banana, essentially, right? And so if it has a disease that it can't overcome, it's susceptible to wiping out all of them because it's the same. It's a clone. They're cloning the bananas.
That's right. And there are people in Australia who are trying to use the immune system, now that we understand it, to engineer bananas that can fight this disease called Panama disease. So
that's what it's called, Panama disease. Yeah, let's
move on.
Wow, that's going to hit some people.
Now, you know, you mentioned chocolate and coffee, things we care about in the developed world, in a rich country like the U.S. But when you talk about calories provided around the world, I think we all know the big ones, right? Wheat, rice, potatoes, cassava, and potatoes. Now, here's what happened. So this picture in the background is a field of dead potatoes, and the one potato plant behind the graphic is disease resistant. It's different from all those dead potatoes by one gene. Okay, and that's it.
One gene.
That's it. So if you could click backwards one.
Yep.
So this is what happened in Ireland in the middle of the 1800s. The Irish potato famine killed 750,000 people. 1 million immigrated, left the country. Many came to the U.S. Pretty much all the Irish-derived folks in the U.S. can blame the potato famine on their presence here. And you'll note that look at the population drop from the peak of the population into the potato famine, which peaked in the late 1840s. Ireland's population has never recovered from that.
Wow. I mean, so there were 8 million people in Ireland. There were more people in Ireland then than there are now. And a massive exodus happened because of this famine. Most of the Irish people here today, maybe we can say the whole Kennedy Empire is thanks to this famine being here, and all your great Irish Americans, perhaps here because of this great famine of a single crop that had such an important impact on the total amount of calories of
That's
the
right.
entire country. And so, wow, to see the population drop in half from, let's call it 1840 when it
Yep.
happened, somewhere around 1845 to 50, all the way to, by 1950, 50 years later, we literally have half the amount of people. Can you
That's
imagine?
right.
Can you imagine losing half of your people?
There are other political issues and economic issues that contributed to that. But I think that if you read a little about the history of the potato famine, the intersection of the famine and the political issues and the economic issues, that's a perfect storm. And there are places today.
Sorry, go.
Go ahead.
It goes back to something we had talked about offline, which is so, I think, important to get across, is that the beginnings of civilization as we know it, from hunter-gatherers into the civilization we know, is based on this domestication of wheat and these crops. And this is where we start to build out mass civilizations. So we know that it's the bedrock of our civilization. We think that it's financial forces, maybe it's monetary forces, maybe it's political forces. But, all the political forces in the world cannot survive a civilization that doesn't have food. When food is running out at the grocery stores, it doesn't matter what your political belief system that you want to put in and checks and balances. If you can't feed the people, you're going to have catastrophe, because that is the bedrock of civilization.
That's exactly right, my friend. All right, so let's go ahead. There's a couple other things that this slide illustrates that I want to just make sure people understand. As we saw earlier on early slide, about 15 to 30% of our crops worldwide are lost to pathogens and pests. And it's important to note that it's after the energy, the sun's energy, the water, and the labor input have gone into those crops, right? And water, if you live in a city, you may not think about this too much, but in the U.S., water table, subterranean water, is a huge issue. We are depleting our water reserves. So this is economically relevant. Worldwide, there's at least $300 billion in losses every year. Most of that is not to giant corporate farmers. It's to small landholders in Africa and Asia and South America. And then I just want people to get a grip on malnutrition. Malnutrition deaths every day, roughly 24,000 a day. That's basically the same as the number who die every day from cancer.
That's profound.
So the last click here is that if we can increase plant productivity by combating insect pest and pathogen losses, we would save land because we'd be more efficient. We wouldn't be losing that 15 to 30%. And we'd create a lot of new water because we wouldn't be wasting that water. So there's human toll and there's environmental toll to plant diseases.
So walk me through sorry, I lost it, how the water factors into this, the water loss.
Irrigation. So if you irrigate...
Just irrigating plants that never survive.
Exactly, they kill blatant life. So look at that.
So you're just wasting the water on
plants.
Just wasting
the water.
Yeah, got it.
All right, so how do we figure this out? So as I mentioned a while ago, crops were domesticated roughly 5,000 to 10,000 years ago, all of our major crops. And humans are smart, so we kept picking the best plants to keep the seed from, and they plant that the next year. So plant breeding has been going on for a long time. Now let's flash forward to the 1860s. The Austrian monk Gregor Mendel, experimenting with garden peas, establishes the rules of genetic inheritance. He publishes this in German in an obscure Czechoslovakian journal. No one reads it. Darwin is sent a copy, but he can't read German, and he never reads it.
Wow.
In 19, Mendel dies. Mendel publishes that in the 1860s. He dies rather unknown. And in the early 1900s, between 1902 and 1905, about 4 scientists in the UK and in Europe rediscover Mendel's work. And they go about using different organisms, plants, animals. They go about proving, again, that his insights were correct. And among those guys was a chap named Dr. Biffen in the UK, in Cambridge, and he started looking at disease resistance. And he noticed that some wheat plants were resistant and some were susceptible. So we'll call the susceptible ones here with the little r and the resistant ones with the big R. So when he crossed those together, the progeny were resistant. So resistant is a dominant trait, right? One copy is enough in the offspring to give you resistance. And that's important because it makes breeding easier. You don't have to go many generations to see it. So in the 1940s, an American guy named H.H. Moore, working for the USDA, I'll give a shout out to the federal government for supporting science. And in North Dakota, he started looking at flax, which is an important crop then for fiber, and flax rust. And you see the photos here. The top panel on the right, resistant parent, bottom panel, clearly susceptible. So a group of friends of ours in Australia figured that system apart, and others of us started working on other plant systems to see if this was generalizable and to use the new tools in the 1980s of recombinant DNA, molecular biology, to clone the genes for these resistance genes. Okay.
So you began cloning the resistant genes. Luckily, it's the dominant. I mean, that was such a blessing that the resistance is dominant.
It is nice. Go ahead. It made life easy.
Yeah, I mean, it'd be like brown eyes versus blue eyes. We know that brown eyes are dominant, and so it's going to take you 2 parents essentially with blue eyes to make a blue-eyed kid. If it's blue and a blue-eyed person and a brown-eyed person, they're going to have the brown-eyed kid.
That's right.
Roughly speaking. And so, you know, I mean, it could have been the other way around where disease-resistant was recessive, and you'd have to work a lot harder to, get these made. So that's
there are some recessive resistance genes. Uh-oh. Right, so as I mentioned, several of us use different systems. And if you imagine pathogens in general, think of your own pathogens. Bacteria pathogens that cause food poisoning have a different lifestyle than viruses that need to replicate inside your cells, right? So plant pathogens are the same way. There are those guys in the upper left-hand corner that grow in between cells, shown in the inset in purple. They're growing along between the plant cells. In the end, they shoot out these fruiting bodies. It's a bit like The Last of Us, and they come out the leaf, and that's where all the spores are. The fungi that you've seen previous slides, they grow on the surface. They inject proteins into the host cell. to convince the host cell to give them nutrients that may replicate. The lower left shows bacteria. It's a cross-section through a leaf. The hollow cells are the leaf cells, and the little clumps right there, those are bacterial colonies. They too sit outside the cell. They inject proteins into the host to make the host give them what they want, which is carbon. And then there's nematodes, the little worms that they penetrate into the root. Then they too, they have a specialized feeding tube that they inject into a host cell and they extract nutrients. So all these.
Sorry.
Go ahead.
Well, I was gonna do a mini side quest, as you mentioned, The Last of Us. I mean, what's your take, being so well versed in this space with that show and its depiction of what could happen?
Well, there have been many epidemics in the human species since 10,000 years ago, right? There have been plague epidemics, Spanish influenza, COVID. So, there are things that could wipe out a bunch of us. That specific scenario I'm not too familiar with.
Yeah, the zombies and all that. Yeah, that's.
Yeah, well, there's a lot of track, but there are fungal pathogens, and in fact, that shows based on the observation that there are fungal pathogens that infect insects and turn them into zombies and change their behavior. You should find find one of the guys that works on that, have them on. All right, so all these different lifestyles, not yet.
Okay.
All these different lifestyles of pathogen beg the question of whether there's different ways to perceive different pathogens, or whether there's a single way that's systematic. And if you go to the next click, there we go. So it turns out when we cloned all these genes, these disease resistance genes, against different pathogens in the mid-90s, we and many other labs, turns out that they all encode the same class of proteins that function inside the host cell. And we call these innate intracellular immune receptors. And based on their protein domains, they were given the name MLR. So I might use those terms in red as we move along. So this this is the main reason that there is a plant immune system. All pathogens, all lifestyles, same perception. Think about the human immune system. It's the same way, right? T-cell receptors perceive pretty much every class of pathogen. Antibodies can detect any antigen from anything. So it's a unified system.
Now, how does it vary to have a sort of singular system that's unified across all plants? So you discover that basically all plants have the same immune system, the same way that all animals have a single immune system. How does that vary from a single point of failure within an individual who obviously has that same immune system? Why is there something that can overcome this entire framework of immunity?
That's a great question. So these MLR genes that encode these receptors, they're hugely diverse in both the protein sequence and in their distribution through individuals of the same species. So if you look at the model plant that we work on, and you genome sequence 1,000 of them, they're very diverse from individual to individual. So obviously, I have to give you one more point. At the first level, one gene encodes a receptor that interacts with and responds to one pathogen, antigen, if you will, right? So you can have holes in the repertoire of receptors. So it is the case that the population is likely to be resistant to everything, but it's also the case that many individuals will not be, right?
Yeah.
And so this is what can give rise to stable, balanced interactions in the wild that are not epidemics. Now, the problem arises when we breed, let's say wheat, for all those genetically complicated traits that we love about wheat, the size of the grain, the flavor, when it flowers, so it escapes the frost. So when you get all those genes selected, you've also selected a subset of the species repertoire receptors.
So
each,
you sort of subset for some unspecified selection of immunity.
Exactly right. And so that's a problem.
So you're saying that there's actually more resilience in non-genetically modified plants.
Well,
You
would say non-genetically modified. So there's more resilience in the population than there is in any single strain or cultivar derived from that population.
Got it. All right.
All right. So now I'm going to tell you the rationale of the immune system. Now that we know the players, so we know two things.
Let's tee it up real high level first.
Yeah, we know two things that we've seen in previous slides. All these classes of pathogens, despite whether they live inside the cell or outside the cell, they deliver proteins into the host cell. And the receptors, when we clone them, proteins were predicted to be inside the host cell. But we also knew, we haven't talked about this, we knew that there's recognition that's based on the surface of the host cell. So let me set up this graphic. On the left, the purple squigglies are a plant cell membrane. Outside that membrane is a gray bacteria. It's got something called a flagella. There are black lines that help it swim. Those flagella can be recognized by a protein on the surface of the plant cell. Stuff happens inside the cell, and disease resistance occurs. That has nothing to do with the genetics or the receptors I've told you about so far. And I'm not going to go into any details. But if this were the case, the plant will always win and there'd be no pathogens. Because the plant is perceiving bits of the pathogen's coding that it can't change. You got to swim. Fungi has to have a cell wall, right? So imagine if in your case you had antibodies to something on the surface of an E. coli, so that that E. coli could never have variation because it needs to have its coat of arms on the surface. So your antibody is always going to win. If that were true, there'd be no disease.
Right.
Okay, luckily, for pathogens, unluckily for us, there is disease. So give it a click. So what successful plant pathogenic bacteria, those gray guys again, outside the cell, they're still tickling the receptors on the cell surface. And that would lead to the cascade of alphabet soup on the left that would cause resistance. However, a successful pathogen, at least for bacteria, injects into the plant cell about 50 proteins, depending on the species and the strain. They're called effectors. They have effects. The effects they have, as shown in the middle here, are to inhibit the resistance signaling. So they shut off the defense, and they're inside the host cell doing that job. So think about the lifestyles issue again. Nematodes, viruses, fungi, they all deliver effectors to the inside of the host cell that help shut down the defense system that the microbe cannot avoid. All right? So now the plant's susceptible. So now the plant loses, and if that were true, we'd have no food. All right, last click. So now the logic circuit is closed by these MLR receptors, shown here with the green squiggly lines on the right. Don't worry about the alphabet soup. And those guys bind the effector directly, truly a receptor, right? Or they sense the alteration in the host cell caused by the effector. Okay? So they reboot the interrupted resistance pathway at the left, and it leads to resistance. So now we have a balanced problem. Now that the MLR proteins, the receptors inside the cell, are recognizing an effector that's trying to do its job to shut off defense. So there's a huge evolutionary pressure on the microbes to lose the gene for those effectors because it's getting them killed. And they lose them a lot. So if they lose that gene, then the MLR receptor is rendered ineffective. So that's the logic of how the system works.
So with that last bit, you said which side is shedding those genes?
Pathogen. The pathogen.
Got it.
The
pathogen is shedding the gene that was turning off the defense, but then failing to keep it off.
That's right.
Because it failed to keep it off, you're leading to my death, so I'm going to shed you, which in some sense gives the plant better resistance over time because it doesn't have to fight it anymore.
Yes, except that the pathogen effectors are redundant. And by that I mean, look at those top two red boxes that are labeled effector. They're both inhibiting the same linear set of steps from the cell surface to the nucleus. So I could lose one of those and still inhibit. Maybe not as well, but I could, right? So this is it's a complex evolutionary arms race.
Yeah, and so it's so beautifully put here, and pretty it's great to grasp it in the sense of these different offensive capabilities essentially going through this protective layer, and if they do happen to leave these proteins, then the effector, the LRR comes in and is able to neutralize that and turn the defenses back on.
Yep.
And so I'm guessing that it's failures in this LRR to something that's, novel, that it can't stop, that makes it non-resistant eventually and die. And so is that where the failure eventually happens?
Right, so if the LRR gets defeated by its effector disappearing from the pathogen population, that's just natural selection, right? That's evolution. If I put a resistance gene into a cell, a single resistance gene, that gene will ultimately lose, right? So now that gene either degrades, it's useless, or its sequence is borrowed and used elsewhere during recombination. So there's lots of ways to generate new diversity in the receptors. Remember, I told you that there's 100 to 500 in any individual plant, and they're sequence related, so they can recombine, and they do to generate new diversity. So it's a constant war.
So they keep up in the defense and they keep up in the offense.
Yeah, exactly. So what do these things look like? So using the power of new structural biology tools, lots of people, I think led by Brian Staskowitz and Gigi Chai in China and others, we now have a good idea. And all I want you to get out of this slide are three things. On the left is what the MLR receptor really looks like. And the different colored blobs are different parts of the protein. Then, in the second step, it binds the effector that's going to turn it on. And the thing goes through an amazing molecular rearrangement from a monomer, so one copy of the protein, float around the cell, to a pentamer. You can see there's 5 in that wheel in step 3. And the gray middle end of that pentamer, if you rotate it, that gray end sticks into the membrane and punches a hole in it. And the plant cell that's infected, that activates the NLR, commits suicide, and that contributes to stopping the pathogen from growing.
So let's maybe take that back then to, I mean, maybe you're going to do it as you go forward, but I was thinking of going back to something like this. Now that we've dove in a little bit,
We'll come right
maybe
back
there's
to
an
that.
example of how our coffee is getting demolished.
We'll come right back to that slide in about 3 slides. All right.
That's why you're the the professional.
So go back one.
Oh.
All right. So these activated receptors, they need to oligomerize into these big machines to essentially create a hole punch that causes the membrane to leak and the plant cell dies. And that plant cell that's dying also signals its plant cells around it to turn their defense on. Now, remember I said at the very beginning, there's no circulating defense. Every cell needs to be able to defend itself. So the way they do that is, typically, they commit suicide that contributes to the death of the pathogen. And they signal many cell layers out from the infection site to turn on defense. All right? And so now there's 100 of these, 200, 500. They're not quite all the same. There's different sub-flavors. And I just wanted to give you just a glimpse of the diversity. Go ahead, one more.
Yeah.
These are simply 5 different immune receptors, MLRs, whose structures have been solved by others. We don't do this stuff. It's too complex for me. Okay, all of them lead ultimately to this cell suicide by punching a hole in the membrane. All right, one more slide.
So the cell basically commits suicide and signals to everyone else, turn your defenses on because I just died. And they try to neutralize it there at the spot. Exactly.
Okay. So that's the immune system. So now we're back to our favorite slide. And what can we do with this? So the 3 that you saw before are the 3 that I'll give you some examples of. Because Brian Staskowitz and Jonathan Jones my friends and co-Wolf Prize winners have actually made, along with several colleagues, some progress on this. So let's go forward now. What kinds of solutions can there be? So here's the tomato wilt. So the tomatoes on the left are market tomatoes, typical market strain, grown in Florida. The tomatoes on the right, and that's a bacterial wilt disease. Bacterial speck disease. And tomatoes on the right carry a gene that is clearly curing them of this disease. So one disease resistance gene that actually came from bell pepper. Bell pepper is a close relative of tomato. They made a transgenic plant, and that's a cure. Now, why? I told you a minute ago that a single receptor will always get defeated, right? Because the bacteria, in this case, can jettison the gene whose protein triggers its function. All right. There's one case, evolutionarily, where that can't happen. What if that bacterial effector gene is also absolutely required for normal bacterial growth? Bacteria can't jettison it, right?
Yeah.
And that's the case. Brian Carney in Brian Staswitz's lab showed many years ago that the effector trigger for this pepper MLR is absolutely required for bacterial growth. And in repeated trials over 10 years in Florida, there's never been an outbreak of a mutant bacteria that escapes this MLR. So this is truly durable resistance. And this is a great example of what we can do studying the genetics of the host and the pathogen.
So,
and go ahead.
So they put a NLR immune receptor. So, you turn the defense off. Then there's the receptor there ready to, fight the invader and turn the defenses back on. If that is, defeated, then we have a problem. What they discovered was there was a way to put a defense mechanism in that layer that could neutralize all forms of bacteria because the bacteria needed that to grow. So they neutralized basically where the bacteria grows from as the target for the defender. I think I got this right. Right? And so that defender is now essentially put the best defender in the world inside that layer after the defenses are turned off. This defender that's able to basically say, I know your kryptonite. I know all bacteria is kryptonite.
That's a good
none
analogy.
of you can essentially get around this one because all of you need this thing to grow.
All of this one species is called Xanthomonas. And so that's a cure. And what's really important about this cure is that along the Gulf Coast, especially in Florida, the way that you fight this bacteria is with a spray called Bordeaux mix. It's a mixture of pesticides, but it happens to contain copper for reasons we won't go into, but the copper is very important for the pesticide to work. So there are places in Florida where farmers are required by law to collect their runoff water because it's got toxic levels of copper in it.
Wow.
Okay, so this, not only does this help save a disease, it helps save contamination of groundwater.
Wow. So, this is way better than what they're using now. Fantastic.
It's way better. And I can tell you that over the years, Toothpaste Foundation has had quite a hard time convincing tomato growers to adopt this and sell it. Even though
I was going to ask, you said that happened a while ago, and they still haven't adapted it, and they prefer to contaminate the groundwater.
Yeah, because of fear of GMOs.
Because of Fear of GMOs. Got it. So they fear the sort of perhaps known effects of these GMOs compared to the definitely known negative impacts of copper
I think
contaminating groundwater.
that's a decent summary.
Wow.
So those are
Very
issues.
intriguing.
Those are issues that society and your food chain have to deal with, right?
Mm-hmm. Mm-hmm.
And so the label there that says Embrapa is a Brazilian funder and agriculture agency, and there are now field trials going on in Brazil. So hopefully they'll find a market for that.
Now, is there a profit incentive beyond? I mean, is there something about the pesticide? Is there increased costs? Is there something in a profit motive scheme that may also be leading them to want to stick to the copper contaminating
But
solution?
I don't think so. I think it's just, I think that the marketing surveys seem to convince the growers that they are better off sticking with the current plan. Now, maybe it's the case, and I'm completely spitballing here. Maybe it's the case that the public doesn't really realize the dangers of the copper contamination, right?
Yeah.
Yeah, imagine if you're a farmer. Imagine if you're a farmer and let's go ahead. You can edit that out. Go ahead. All right, so look, earlier we talked about new epidemics and how they spread and how that spread leads to or contributes to societal unrest. So on the left is our old friend, wheat stem rust. You can read about this in documents from the Roman Empire. And there have been two other outbreaks of this in the early 20th century. The latest one started in 1999 in Uganda. Panel 2 here is a map of East Africa. You see the Arabian Peninsula there and the Red Sea. The small lower left black box is Uganda. And that's where this particularly virulent disease-causing strain came up in 1999. In 10 years, go over to 2019, you see how far it's spread. Now, I'm sure that most of your listeners are aware that there's horrible famine in Sudan and Eritrea. They're right in the middle of that. So again, we have a disease, terrible famine, causing huge losses that overlaps with breakdown in societal and governmental structures. So lots of people had started working on this 25 years ago. And the solution this time is not a single receptor that hits an absolutely indispensable bacterial effector. In this case, it's a stack of 5 genes. So let's go forward one. So this is work of folks in Australia at CSIRO, McAuliffe and colleagues, and people at the University of Minnesota named John Stephenson, his group, Two Blades, and many others. So on the left picture are wheat plants. The dead ones are susceptible parents. The live ones carry a stack of 5 different receptors. Each is different, but each recognizes effectors from this new strain of wheat stem rust. Okay? And on the right-hand panel, you see leaf blades from that experiment. Controls, obviously 6, sick as hell. The lines with the 5 gene stack, healthy. So 5, think about mutation frequencies. The likelihood that you mutate all 5 effector genes to escape this big 5 line is a lot lower than the likelihood to escape 1, right?
Yeah.
Can happen, but it's just a lot lower frequency. So this at least buys time. So that's another kind of solution to a big problem. Go ahead. All right, now we're back to our old friend, potato late blight, the Irish potato famine. Jonathan
Still
Jones,
happening.
still happening. Still a problem in Europe,
Still
huge
running
problem in Europe.
around. I mean, these are just species. They almost have their own character to them. They have their certain way of attacking. They have their certain lifestyle. They might have environments they like to live in. I mean, these things are crazy. I mean, this thing has been around for hundreds of years now. Like it was, yeah. I mean,
Yes.
wow. And so it's still roaming the planet. It's interesting to think of them like the organisms that they are. But we don't think about potato late blight the same way we think about lions or something.
Right.
But in essence, it
But
does
we
have the
should.
same survival
We
mechanisms.
should. You're more
Yeah.
likely to die. There's more deaths due to potato late blight than there are from lions every year, if you consider malnutrition, right?
Yeah, no, that's a way to look at them. I mean, these things are more dangerous than lions. They really are, bacterial predators. Are they bacteria? Sorry,
So this is not a bacteria. This is a
it's a virus.
funny organism called the oomycete. It's
Oh,
actually
that name. Yeah, I saw that. I don't know much about them.
they're commonly miscalled fungi because they look like a fungus, but they're most closely related to algae. So
Got it.
evolution. So anyway, here's the result. Jonathan Jones in his lab and Two Blades and many other co-workers and colleagues, they stuck 3 receptors into the dead potato, and you see on the right the result. So these are going to be, these are hopefully going to hit the market in various places around the world soon.
Wow.
So, so the take-home message on the next slide is you've already seen the top. Plant health is human health, environmental health. And what I want you to take home from this is that the fact that we now know the molecular machines that comprise the plant's immune system, including the immune receptors we talked about today, provides raw materials for engineering solutions to provide healthier food on less land using less water and chemical inputs. So that's the message. That's why it matters.
Yeah. It matters tremendously, and unfortunately, unfortunately, but it's so hard to, I think, make it hit home to people who are doing fine, who can afford the price increase of. I mean, coffee would probably be my breaking point. But it's just until calamity strikes, it's hard for people to realize the severity of these kinds of issues, and so. Thank you for your work and everything that it's done to illuminate that and help solve it. I mean, you're keeping humanity alive and
Well,
civilization flourishing. I shouldn't mention that. I mean, that's a huge part of it. Is that it's a bedrock of civilization.
and I think, it's not just plant diseases. Egg prices, right?
Mm-hmm.
Increases in egg prices in the U.S. are directly a consequence of bird flu epidemic.
Mm-hmm.
And the
Yeah, and I
fact
think
that,
people that one has really hit home for a lot of people. I
yeah,
hear people. I was just at the barber shop, and they're like, "Man, I don't want to go get takeout breakfast because the prices of the egg and your takeout it just made the whole price go up." And so now he's like, "I don't know if I want to tell my girlfriend that I'm getting food because then I'll have to, spend over $30-$40 just for us two." And it's just like so bizarre. He's like, I guess I'm just gonna eat at home, or maybe I'll eat something and not tell her. I mean, that's awful. It's ruining relationships. But more importantly, closer to home, it's the awareness of something as vital as eggs, and the causes that have brought it to light. And so I think that's a great example that's actually starting to impact people and make them understand something's going on. But even there, it seems like it gets thrown off and cast away as political. You know, I think they hear about the bird flu, but people are
well,
still like, maybe the
but,
president has something to do.
we've had to kill millions of chickens So if you kill millions of chickens, you're losing 5 million eggs every day, or 3 million eggs every day. Each chicken lays 2 or 3 eggs. And so that's supply and demand, man. Price is going to go up.
That's right. Fantastic. Well, I really appreciate you coming on board. Anything else you want to leave people with? I know we want to talk future, anything topical. I mean, what's the last thing you want to leave people with before we have to wrap this up?
The last thing I want to leave people with, based on the conclusion of my last slide, is that whether you want to have organic food, whether you don't care how your food was grown, whether you pay top dollar at the local farmer's market, or whether you buy it at a large, big supermarket chain, the way that we grow your food has huge consequences on the environment in terms of chemical inputs, water use, and water use is huge. And by understanding the plant immune system and understanding dynamics of how disease-causing microbes move through the environment, and then by strategically developing resistant crops using either GMO or CRISPR technology, we can quickly overcome certain diseases that, while they may not have a huge impact in a rich country like the U.S., they're serious business in the developing world. And it's my view that we as scientists and we as quite privileged Americans, that we continue to use our technology, stretch it into the future, and deploy it as cheaply as possible around the world to make the world better and safer for everybody.
I mean, that should be the true American spirit, and in the human spirit as well. Dr. Dangl, thank you for coming aboard.
Thank you, Andre. My pleasure.